Hey,
Hi,
Andre.
Aaron. Okay,
Good to see
sound
you.
check. Positive. Very good.
Sounds great.
How's it going?
Thanks for joining. It's good.
Oh, thanks for the opportunity. That's actually pretty amazing. Yeah, somehow got a notification through the through Stanford newsletter. Actually, not me. My wife. She was also a postdoc at Stanford, and she knows a few people. And she somehow she informed me that this exists.
Oh, great. So it's in the Stanford newsletter?
Was it a newsletter? Maybe it was some other newsletter people sent around. Okay, you should join this. This is a very interesting series. How do you usually advertise for the series? Is this more internal, or because I would definitely
No,
let people.
it's just word of mouth.
Word of mouth, yeah. So I will let people know here what's going on. I think they would be very interested in this. You seem to have a very good setup, or very good speaker list.
Yeah, please spread the word.
Hello.
Hi, Kalanit. Thanks for coming.
How are you?
Good. So Andre and Kalanit, would you like to test your slides out first? Maybe Andre first.
Okay.
Your wife is still a postdoc at Stanford, or she was?
No, she went became a research scientist at the Allen Institute.
Okay.
Just across the street from where I work, so it makes commuting easy.
But you were at Stanford, right?
Yeah, we both were. Yeah. So
got it. Okay.
Got to know each other in Cards Lab.
Yeah.
We both moved to Seattle. So you see this here? Does this work? You see by...
Yeah. Yep, looks good. You want to try the next slide? I don't see the... Oh, yeah, there you go.
It's a Java update notification. Oh my gosh, that's not good. Okay, we bring this back. Yep. Okay.
Yeah. Looks...
Okay, and that should be video showing some GK stuff here.
Yeah,
right.
Perfect.
Great. Okay, I'll stop sharing.
Thanks. You want me to try?
Yes, please.
You don't have like a... okay, let me see. Okay. I still can annotate on it. Okay, good. Can I just try?
It says underneath subtitles aren't currently available. Do you want that there?
No.
Okay. I usually use like a Zoom feature, which will do like a live transcript. Actually, I can start that now. It helps.
So what do where should I do that in the Zoom?
No, I'll do that. I'll do it actually now. So just as we're talking, it'll convert it to text just so people can read along. But it seemed like maybe you had it in your PowerPoint setting to have a transcript.
So how do I change that?
That I don't know. Hi, it's Along. How are you?
Pretty good.
Morning.
Morning.
Okay, figured it out.
Oh, great, great.
I think so. I think I clicked something by mistake.
Oh, okay.
Perfect.
So, Kalanick, your talk will be second, and then Andre's will be first.
Could you just mind if I share the screen again just to see how
Please,
it shows
yes.
up?
Sure, yes, please.
Okay. Because it shows
Okay.
only the presenter view now. Why is
Yeah,
that?
no, but that's okay. So just go back to you just have to swap the displays. You could do presenter view, but then you just do what you had, and then then
Yeah.
swap that.
Okay.
Yeah.
Okay. Okay, got it. Okay.
Yeah, that looks good.
Okay, now stop sharing.
What's the weather down there in California?
I'm on sabbatical in Shanghai, so
Oh, and yeah, good morning to you too.
The Shanghai-based meeting.
yeah.
Yep. Nice.
I was
Actually, it's
wondering about the time zone. Now that explains a lot, actually.
I'm usually up at all hours of the night anyway, but yeah, the time zone is a little different. Now it's very hard. Now China canceled most flights from the US to China, and then US responded and canceled most flights from China back. So it's hard to leave, and so it's a little bit tricky right now.
Oh my
And
God,
you-
the postdoc is supposed to come back.
Yeah, and then yeah, it's complicated.
Do you have to go
But
to
with,
Singapore also?
yeah, you could do that, or Dubai or whatever.
Yeah.
But then there's another thing where if you get COVID, you can't come back for like 6 months or something like that. So it's a little bit tricky.
Okay.
It's Chinese New Year anyway, next few weeks. So it's like Christmas in China. So it will take maybe flight come back next month. I bet.
Hope so. Yeah.
Yeah.
I like your Stanford sweatshirt, so.
Oh, thanks.
Oh,
This is like
It's
is
a
a
Neuro
Zoom.
Zoom sweatshirt. Okay.
Zoom. Yeah. Yeah, this is a
yeah.
rare one. There's only a few of these around. I think only 2 in China. Zolong, you have one of them.
Yeah. I think New Year is Tiger Year next year. Yeah.
Yeah, we should make another. We should make another one of these shirts or something this year. We've had, Kalanit, we've had a few psychology department talks from different universities here. So we'd like to get more diverse speakers.
Right.
How does the Gold conference play out when you're in China? Is that the reason why you need to come back? I thought your chair.
The Gordon conference?
Yeah. It is.
Yeah, people keep reminding me of that. Yes, that's in July. Yeah, so I do have to come back at some point for that.
You know
we'll
if
see.
it's happening?
It was supposed to. They say it is so far. We'll see.
Okay.
It all depends on the situation. I was hoping it was supposed to be in Hong Kong, but then they moved it to California.
All right.
Which is less exciting. But yeah, July is a long time away, so hopefully things will.
Yeah, hopefully we meet there. I also submitted an application for that thing with the actually the topic that I'm presenting today.
Oh, great.
It's
okay.
not really published yet. So we hope to have a bioarchives paper out at the end of the month and then move it forward to submission stage. It was a very long project. So I hope I convey the message in 20 minutes.
Great. Well, there's editors who watch this and things, so that's good. I like bioarchive also because you just submit it. The next day it's online. You don't have to worry about any long review process.
That's done. Yep.
People read the bioarchives. I feel it's much more democratic. It's much more accessible to everybody.
Yeah, it's my favorite journal now. We have papers that are
Very-
under review that I've forgotten about it, but bioarchive is good.
Okay.
All right, Zilong, should we get started?
Sure, go ahead. Yeah, should.
Okay, all right. Hi, everyone. Thanks again for coming for another week of NeuroZoom. We have great talks from Andre Berndt and Kalanick Grill-Spector. Before we get started, just as a reminder, next week, because of Chinese New Year, we'll take a week off, so no NeuroZoom talks. But then, so no NeuroZoom talks next week. But then we'll be right back at it with exciting science, Qianchu Yi, and you can never pronounce her last name. I'll just say Jenya K from Northwestern University will be giving a talk on amazing proteomics tools. So tune in then. And lots of slots available for March, April, May, if you'd like to present your latest research. So please sign up. I'd love to hear from you. Okay, Zilong, do you want to introduce the first speaker, Andre?
Hey, great. Thank you. So it's my great pleasure to introduce Andre. By the way, it's the last talk between the bull year, Chinese bull year, between holiday. Everybody here is very excited to have a week holiday. So Dr. Andre Berndt has his PhD from Hamburg University. Actually, Andre has devoted his career, a long-time career, over near 2 decades to develop all kinds of blockbusting tours to dissect neural circuitry like optogenetics. So after the PhD in Germany, he come back to Karl Dazzler's lab in Stanford to keep working on his tool, develop all kinds of tools, engineering the optogenetic tools to control neural circuitry by light. So after the postdoc, he started his own lab in University of Washington, where he has more diversity tools. So today he'll talk about the massively parallel, also high throughput engineering for optogenetic biosensors. We're excited to have Andre to bring us more exciting tools to dissect neural circuitry. So welcome, Andre. Take it away.
Yeah, thanks so much, Zilong. And thank you, Aaron, for the opportunity to present here. Okay, let me start sharing the screen right now. Okay, yeah, I have only recently heard about this seminar series, but it became a permanent entry in my calendar. And I'm looking very much forward to hear more exciting talks in the future. Yeah, so basically everything I'm going to present here is not published, so therefore also a great opportunity to spread the word of our recent research here at UW. Okay, I want to give you a very brief overview of my previous work, which directly motivated my current research here at the University of Washington. As a graduate student and later as a postdoc, I mutated the light-activated ion channel rhodopsin 2 and engineered new biophysical properties. For example, I changed the ion selectivity of channel rhodopsin from cations to chloride, which resulted in inhibition of neuronal activity under light application. And I also generated faster variants of channel rhodopsin to enable light-induced action potential generation at frequencies up to 200 Hertz. And of course, this is now ancient history. What's really important here is that in about 10 years of research, I only tested about 600 mutated channel rhodopsin variants. And considering the vast sequence space of proteins with billions and billions of possible variants, this represents a very small subset. So I set out to make the engineering of protein-based tools for neuroscience research a bit more efficient. So I also changed my focus away from optogenetic actuators such as channel rhodopsin that control neuronal function to optogenetic sensors that monitor neuronal activity. So the prime example here of a sensor protein is, of course, the calcium indicator GCAM6, which is a fluorescent protein that fuses a calcium binding domain with a fluorescent reporter, in this case, circularly permuted GFP. We can express these tools in specific subsets of neurons and monitor changes in calcium dynamics in real time and with cellular resolution using fluorescence microscopy. So in neurons, calcium dynamics, of course, are strongly correlated with electrical activity, such as action potential firing. So with using GCAM, we can optically observe action potential firing in real time in intact neuronal circuits. We can do this even in vivo, as shown here in this example, where GCAMP is expressed in principal cells in the layer 2-3 of a cortex of a live mouse. The fluorescence here is detected with 2-photon microscopy through a transcranial window in the skull. And of course, the widespread use of GCAMP in neuroscience has now motivated the development of a whole array of methods to detect dynamic fluorescent signals in live animals. We just saw an example of in vivo 2-photon microscopy through transcranial windows. This can be further enhanced by using green lenses to reach even deeper brain regions. And we can also use fiber photometry to read out light fluorescence in freely moving animals. We can even mount mini microscopes onto rodent heads to read out fluorescent signals with cellular resolution in freely moving animals. So we have a wide variety of hardware available to us, and what we need to make this even more useful are a variety of sensors that can be used in combination with these methods. And of course, there are hundreds of biochemical compounds in the brain which are involved in information processing. We have neurotransmitters, modulators, and peptides, for which our colleagues already developed fluorescent sensor proteins in the past. One great example here are sensors based on G protein-coupled receptors, or GPCRs. 2.5 years ago, two groups, Lin Tian at UC Davis and Yulong Li at Beijing University, independently fused fluorescent circular permuted GFP to the third intercellular loop of the human dopamine receptor 1, which undergoes large conformational changes upon dopamine binding. In these fusion proteins, the dopamine binding now does directly translate to an increase in fluorescent signal. And consequently, with these dopamine sensors, we can now detect endogenous dopamine release using the imaging techniques that I just showed you. The good thing here is that many neurotransmitters, modulators, and peptides signal through GPCRs. So with the same engineering principles, we can now hypothetically engineer sensors for many more neuronal signaling molecules. However, fusing CP-GFP to an GPCR is really only the beginning of the work, because many of these sensors require additional engineering to gain the signal amplitudes and sensitivity that's necessary to be detectable in vivo. For example, for GCAMP, it took 12 years of additional protein engineering to reach that point. So these GPCR-based sensors also require additional engineering or mutations in key residues. This is demonstrated here for D-Light 1.1, which comes from Lin Tian's group, and GrepDA, which comes from Yulong Li. The linker residues that connect the GPCR to the CP-GFP are crucial for the signal amplitudes, and both groups mutated key residues here and tested hundreds of mutations to find the ones with the largest signal amplitudes, and I just ordered here from left to right individual variants according to their signal output, right? But many more variants could have been tested. For example, in D-Light up here, 4 residues within the linker region were mutated, marked by an X. With 20 possible amino acids at each site, 160,000 variants are possible. But with the current methods, it would be very laborious and resource intensive to test them one by one. So my lab sees a large benefit in increasing our testing capabilities to test as many variants as possible to identify the best ones from protein libraries in an unbiased directed evolution approach. We can create protein encoding DNA libraries very easily using standard PCR methods. The trick is to separate these plasmids and test these variants individually. And a necessity here is that we want to use mammalian host systems that are physiologically close to neurons. And here specifically, we use a type of mammalian cell cultures that will express only one variant per cell. So once we achieve that, we seed our cells on microarrays where these cells are physically separated and where we can screen hundreds of these cells in parallel. When we stimulate sensor-expressing cells, we can analyze the fluorescent signals of hundreds of these cells simultaneously and identify the ones expressing the best sensor variants in real time. We can pick up these cells from arrays to retrieve their genetic content content and move these new sensors either into downstream neuroscience applications, or we return these variants back into our screening pipeline for additional engineering. And a trained person can set up these experiments very easily and test 10,000 variants in one day, which is a significant improvement over traditional approaches to sensor engineering. So how does this all work? First, we have to ensure that each of our mammalian host cells only expresses one sensor variant from our DNA library. So the problem is that when mammalian cells get transfected with plasmid DNA, each cell takes up more than one plasmid. That means when our DNA contains a mix of mutated variants, each cell would express several of these variants, which makes their independent testing pretty much impossible. So to solve this problem, we use a genetically modified PEG-293 cell line that has a recombinant locus in its genome. So the plasmids that we're using for the library generation don't contain a promoter, so they cannot be expressed by themselves, but they have to be integrated into that specific locus in the HEK cells by recombination. But there's only space for one plasmid, and the process is not reversible. That means only one plasmid can be integrated and expressed per cell, even if this cell takes up more than one plasmid. We demonstrated that here using a DNA library containing only two variants, a red and a green fluorescent protein, that were transfected at the same time. And as you can see, the majority of the cells only express one of them. So the next problem is to separate these cells, because cells in culture tend to grow together, which would make it very difficult to identify individual sensor responses from them. So here we use a microarray made from Pdms that contains 5,000 microwells, each with a 30 micrometer diameter, which is really just big enough to hold one individual cell. And these become physically separated. We cast the PDMS arrays from a silicon mold that contains several dozen of these arrays. We cut them out and place them into 24 well plates and growth media. And after the library transfection of our host cells, we seed the cells onto the arrays and image using standard epifluorescence. The imaging itself and the data analysis is now fully automated and happens in real time to quickly identify the best variants on each of these arrays. Here's a better view of one of the 24 well plates on our microscope. We use a micropipette that delivers drugs or compounds to each well to stimulate these sensors. And here on the right, you see a video of an array at 10x magnification, where we see that about 600 cells expressing a dopamine sensor library that's being stimulated by dopamine. You can see how they increase their fluorescent output upon dopamine application. And we can screen the entire 24 well plate and easily test 10,000 cells or more in just one attempt. Again. Dopamine fluorescent output increases, and we can do the analysis immediately. We can analyze the signals in real time, rank them, and store the exact position of each cell on the arrays. Here on the left, we see the individual traces of each of these 600 cells upon dopamine stimulation. We can then pick up these cells with the highest signal output using glass micropipettes. The graduate student in my lab, Michael Rappe, who has been realizing this pipeline in the last couple of years, made a quick demonstration video for the cell picking. So the cell that he marked to pick up, here is circled in red. The micropipette is coming in from the right. It's controlled by a micromanipulator. And very quickly, we can just move the micropipette down, apply negative pressure to the pipette, and pick up the cell from the array. It's picked up. Once the cells are picked up, we can run RT-PCR to convert the underlying mRNA that encodes the sensors into DNA. Here on the right, you can see the DNA bands from 16 dopamine sensor variants that were retrieved from 16 individual cells. We can cut out the DNA bands, sequence them, and clone them back into mammalian expression vectors for further characterization of these variants in mammalian cell populations or in neuroscience applications. So we had all of these fundamental pieces in place about 2 or 3 years ago and did some initial quality control. Michael transfected our host cells with 3 known variants of the dopamine sensor D-Light, that is D-Light 1.2, D-Light 1.3a and 1.3b. He then seeded these cells on separate microarrays and stimulated them using dopamine. Here in the middle are the signal amplitudes from individual cells. Each dot represents one cell. And the figure here on the right shows a histogram of the individual signal amplitudes for each of the 3 variants in cell cultures. The good news here is that you can identify the variants based on the average amplitude in a cell population. But the problem was that the spread of the signal was relatively large, with a number of cells being outliers. That means that the cell-to-cell variability was relatively high, which is a problem when each variant is expressed in only one cell. So if there's a superior variant that would produce a high average signal, there's the risk that we could miss it because it could produce only a weak outlier signal on our arrays, or vice versa. We could identify a cell with a strong signal that is actually an outlier from a variant that will produce only weak average signals. So that was a problem, and we went to work and optimized pretty much every aspect of this engineering pipeline. I summarized the major points here on the left. But I will not go into the details of what Mike did in the last 2 years to optimize the pipeline, which was a somewhat long and often frustrating process. But in the end, it was also very, very successful. And here on the right, I compared the variability of our mock D-Light library for the known dopamine sensors before and after optimization. And we use the coefficient of variation as a measure for quality control here, which is basically just standard deviation divided by the average signal. And each of these dots here represents hundreds of cells of each sensor variant on arrays. So it's a population analysis. So before the optimization, the variability of our signals was up to 30% of the average signal. And after optimization, we are now down to 10%, which is a very, very good value for this type of pipeline. So with the optimization done, we wanted to test how efficient our platform now can engineer new sensor variants. So we used the dopamine sensor D-Light 1.1 as the first framework for optimization. And similar to previous studies, we targeted 4 residues in the linker region that connects the GPCR domain with the GFP. Remember, only a few hundred mutations have originally been tested, but 160,000 are possible. So we built the library and plasmid DNA using randomized codons and NK for these amino acids, for these 4 amino acid positions. So NK is a degenerate codon and can encode pretty much all amino acids. Here in the middle is the summary of the sequencing of 25 samples from this library at 2 amino acid positions encoded by 6 nucleotides. N encodes all the 4 nucleotides, ACTG, and K encodes TNG. And as you can see, the distribution of these nucleotides is relatively even, and we are confident that our library really represents all possible mutations. So after expression of this library in hex cells, we screened more than 1,000 variants in just under 5 minutes. Here on the right is the result from our very first screen, where we stimulated with 100 micromolar dopamine and screened more than 1,000 cells in less than 5 minutes. The signal output from individual cells is sorted from left to right. We then quickly realized that we don't have to screen for maximum signals. The 100 micromolar dopamine that we applied initially is far higher than physiological values of dopamine in the brain. So we then started to test with 500 nanomolar dopamine, which is far closer to physiological levels, and ensures that we identify variants that work best under these conditions. So far, we have tested more than 14,000 variants, which is less than 10% of the library. But even within the small subset, we have already identified several new sensors that outperform the initial framework D-Light 1.1, which demonstrates the efficiency of this platform. The figure here in the middle shows signals from individual cells on arrays, which all outperform D-Light 1.1. We tested 2 variants, 3A and 3G. That were picked from these arrays, cloned back into expression vector, and we tested them in mammalian HEC293 cell cultures and confirmed that they have a larger signal than the initial framework D-Light 1.1 under low dopamine exposure at 10 and 100 nanomolar dopamine with improved EC50 values. This means that these 2 selected variants are more sensitive than D-Light 1.1. Next, we tested the function of DLIT3A in mouse brains in collaboration with Michael Brooker's lab here at the University of Washington. And DLIT3A has been expressed in nucleus accumbens shell and imaged in acute brain slices using 2-photon microscopy. And as you can see, we get robust and concentration-dependent responses under external dopamine application. These are very early experiments that confirm the function of the sensor, and more experiments are currently planned. In a similar approach, we engineered the prototype sensor M-Light based on the mu opioid receptor MOR. This framework already existed, but produced only weak signals upon opioid stimulation. Similar to the dopamine sensor, we first targeted 4 linker residues between the mu opioid receptor and the GFP by random mutations. And we screened first 8,000 cells on the first day under 1 micromolar Dembo application. Dembo is a synthetic opioid. We identified the variant M-Light2A, which outperforms the initial prototype when expressed in mammalian cell cultures and stimulated by 500 nanomolar and 100 micromolar Dembo. Based on M-Light2A, we then created a second library targeting the intracellular loop of the mu opioid receptor with the goal to shift more of the sensor population into an active state when opioids bind. So our initial screen identified several variants that already outperform M-Light2A. Shown here is the average fluorescence of a variant that we called M-Light3A in comparison to 2A, and the initial prototype we see improved response signals. So far, we have tested almost 30,000 variants for both libraries, and the engineering is still ongoing. We did some additional characterization of these new opioid sensors in hex cell cultures to confirm their function. For example, after washout of Dembo, the signal response goes down, confirming the sensor signal is reversible. We also used endogenous opioids, such as met and neurodevelopment, to stimulate sensor responses. Both of these compounds are the native agonists or endogenous agonists for mu opioid receptors, and the sensor is able to detect them. Additionally, we applied the opioid receptor antagonist naloxone, which reduced sensor responses and can be used as a negative control in experiments. We also generated a loss of function version of the sensor by mutating the key residue aspartate 147 to alanine. This mutation has the same baseline fluorescence as M-Light2A, but does not respond to agonists. So consequently, this variant can also be used as a negative control in experiments. So interestingly, the sensor has relatively similar selectivity as the native mu opioid receptor. Although enkephalins are thought to be the primary agonist for mu opioid receptors, it is known that they also bind other endogenous opioids such as endorphin and dynorphin. Consequently, the M-Light sensor also detects these compounds. But interestingly, our sensor is not very selective for exogenous opioids such as morphine and fentanyl, which usually strongly bind to mu opioid receptors. But in this case, it's okay because we primarily built the sensor for detecting endogenous opioid release. But in our next step, we aim to engineer sensor variants with very specific ligand binding selectivities, for instance, detecting metenkephalin, but none of the other endogenous opioids. And here we can take advantage of the fact that we can apply several ligands during the screen, shown here. So we will target residues in the ligand binding pocket of the mu opioid receptor domain of our sensor, and then screen on our arrays, take advantage of the fact that we can apply several ligands during our screen. And shown down here is an example of a selectivity screen on Pdms arrays with 3 consecutively applied compounds. Here on the left, we first apply morphine, then metenkephalin, and then naloxone. And here on the right, we first apply beta-endorphin and then metenkephalin. And using these paradigms during library screens will allow us to identify variants that have a very desired selectivity profile. So last but not least, I want to share some very preliminary results from testing the opioid sensor M-Light2A in vivo using fiber photometry. Our collaborator, Michael Brooker, published a paper last year that measured the activity in enkephalin releasing and MOR positive neurons that project from the dorsal roughly to the nucleus accumbens. So these neurons here in this experiment express the calcium indicator GCAM and presumably release enkephalin when the animals consume food. They placed a light fiber into the nucleus accumbens, where these enkephalins are released, and let the food-deprived animals consume food while measuring GCAM activity. So remember that these neurons also express the mu-opioid receptor, which inhibits neuronal activity when enkephalin is released. And consequently, our colleagues measured a drop in neuronal activity, which disappeared when the opioid receptor antagonist naloxone was applied. This is an indirect indication that MOR, mu-opioid receptors and opioids mediate that effect. But now with an opioid sensor, we are able to measure that response directly. And here on the right, you see the same experimental paradigm, but instead of a calcium indicator, our colleagues in the Bruker's lab expressed the opioid sensor in the enkephalin neurons projecting to nucleus accumbens. And interestingly, the sensor picks up a signal when the animals consume food, and the signal disappears under naloxone application. And again, this is very preliminary result, and more experiments are currently planned to confirm the function of these sensors in vivo. But I hope I could convince you that our pipeline is indeed able to engineer new sensors very efficiently. So with that, I want to thank the students in my lab, which have done all the hard work. Michael Rapley, seen here, has been working on the pipeline since when he joined the lab 5 years ago. And thanks to his dedication, we can now see the benefits and successes of this project. Justin Lee and Sarah Wade joined the lab more recently and have been instrumental in helping to optimize the platform. They have their own project based on this platform that I could not share with you today, but I hope we can publish those results very soon. Several very talented undergraduate students have been critical in supporting these various projects. I want to especially thank Amanda and Justin, who started graduate school or will start graduate school in the fall. I also want to acknowledge and thank our various collaborators and funding sources. And of course, without these significant contributions, none of these projects would have been possible. And I also want to thank you for your attention.
Great. Thanks so much, Andre. It's an amazing platform. I saw Tim. Did you raise your hand? Or just?
Nope. Sorry, my bad.
Go ahead. Oh, okay. All right. So I got my first question, Andre. So it's amazing such an automated platform can screen so many sensors. But for the beginning, so how you for example the dopaminergic opioids, how do you get such big libraries by some rational design approaches or random evolutionary approaches? How do you start?
So we have to make some decisions. So it was already known from the previous work from Lintan and Yulong Lee that, for instance, these linker regions that connect CPGFP to the receptor are very crucial for the signal output, right? So they can certain mutations can even generate negative signals, right? So signals can go down or they can go up. So that was an easy target. So there's some rationale in selecting the residues, right? And then we can basically just apply these they're very standard PCR techniques to target a small subset of residues and just randomly mutate them. So if we target one residue, then we get 20, a library of 20 mutations, right? If we target 2, then we get 400, 3, 8,000, 4, 160,000. So the number grows and grows and grows, right? But if we which makes it then even harder to, even with high throughput methods, to screen all of them. We have some ideas to actually incorporate FACS screening as well. So there are a lot of sensors that actually don't respond at all. So we could potentially kick those out with FACS screening. So even going to 5 residues, 5 target residues would allow us to screen potentially millions of variants. But again, there's always a rationale. Okay, where do we want to mutate? And it's mostly structure guided or empirical knowledge. So for instance, for the ligand binding pockets, there's a lot of, there are a lot of structures available that show us, okay, where do ligands bind? This is DAMGO here in the ligand binding pocket of the mu-opioid receptor, right? So we just know what are the key residues that are involved in this, right? And these are the ones that we can potentially target. So we pick a few of those, and then we have like a few thousand variants you can very efficiently screen. So it's basically both. It's high throughput, but definitely you need a rational as well to make some kind of informed decision.
Great. Yeah, amazing. Okay.
So I'm confused. You're saying you mutated the link region between the CPGFP and the receptor, not the binding pocket, right?
Yeah, in our first try. So this was, again, we spent a lot of time in building the pipelines. We picked some of the lower hanging fruits. So it was already known that the linker regions are important from the previous papers. But just a small subset of these variants could be tested, just a few hundred, and we just wanted to show, okay, we can screen more of them, like several thousand or 100,000 if we continue doing this, and also find variants that the previous work probably missed. So in the beginning we keep it in the linker region, but as I said, we want to extend it to further regions. So intracellular loop 2, for instance, in these receptors is very important. That's also empirical knowledge in the field that intracellular loop is very important for shifting these GPCRs from like a non-active state to an active state, right? So a larger amount of the molecules can be shifted into an active state when residues are mutated there. So we just applied that knowledge to generate a library in intracellular loop 2, targeting residues in intracellular loop 2, and again proved to be at least not wrong to do that, because we found a variant, or at least one variant, potentially more, that have also increased signal output. So there's a lot of knowledge, molecular knowledge, structure-guided engineering, informing these type of decisions where to mutate.
So if I may, can I ask another question regarding your in vivo photometry experiment? So, the opiate release in response to feeding, is that before feeding presented to the animal? On the right side, C, figure C.
Yeah. So the vertical line here. So this is the food intake, right? And there's an expectation apparently. Yeah. So before the food intake, we see this increase in response, right? Yep, that's correct. Yeah.
Then how do you explain, like, after food intake, there's a reduction, there's a recover to the baseline? That's why.
Oh yeah. So then the subsequent effect of the enkephalin release would be, the idea is that this then deactivates these neurons, but this obviously takes some time. So here we basically just see the enkephalin release, and over here we see the then subsequent inhibition of the neurons. At least that's the explanation for now. So that's the hypothesis. Of course, more experiments are being done and planned, but for now that's the leading hypothesis. Yep.
Okay, thank you.
Great, thanks, Jiping. Is any more questions? Okay, still audience can put your question on the chat board. Andrew could answer the question afterwards. Okay, Aaron, we should move to the second talk. Thank you, Andrew. Great talk.
All right, thanks, Zilong, and thanks, Andre, for that great talk. Our next speaker is Dr. Kalanit Grill-Spector, who is a professor and the chair of the Department of Psychology at Stanford University, and one of my colleagues. Dr. Grill-Spector received her undergraduate training in Israel in electrical engineering and computer science at Ben Gurion University, then did a PhD at the Weizmann Institute, then for a postdoc came to the United States at MIT, and then moved to Stanford, where she's now a full professor and the chair of the department. So her research focuses on studying how humans recognize things like places, objects, faces, stuff like that. For us, recognizing things is very quick, within milliseconds, but the basis of this is unknown, and her lab aims to discover those mechanisms. And to do this, she uses functional brain imaging, fMRI, structural brain imaging, diffusion-weighted imaging. She uses computational techniques, behavioral techniques, all in humans to study the neural mechanisms underlying visual recognition. She recently had a really cool study where she used fMRI to discover how adults who played a Pokemon game as a kid had a specific brain region in the visual cortex that was dedicated to recognizing those various Pokemon monsters. So like Bulbasaur, Snorlax, Venusaur, Charizard, ones like that. Pikachu. She also studies how visual perception develops in young children, and then how that may progress into throughout adulthood, and even how there are similarities and differences in visual perception between populations. She's won many awards. She's received a Human Sciences Frontier Fellowship, the Sloan Fellowship, the Klingenstein Fellowship in Neuroscience, all the major neuroscience awards she's won. And she's an amazing, famous scientist, and it's a real honor to have her present her latest research here. Thank you for joining.
Thank you, Aaron, for inviting me and for this very kind introduction. I'm going to talk at a very different level than Andre, so bear with me. So what I'm going to tell you today is about research in my lab studying how visual cortex develops and improves visual recognition. And this research has been done by a large team of scientists in my lab who are shown here. And in particular, I'd like to highlight the contribution of Marissa Nort. She's my postdoc, who's the first author of much of the research that I'm going to be presenting today. So recognizing faces and reading words is important for our everyday lives. And interestingly, our ability to read and recognize faces improves over childhood development. So for example, if you measure how accurate kids read words or pseudo words as a function of age, you will see that this ability significantly improves from age 5 to 17. Likewise, if you measure how good people are at recognizing faces, whether adult faces or child faces, you will see that children also significantly improve from age 5 to 17. So the question that we are asking in these studies is, how does visual cortex develop during childhood to enable improved visual recognition abilities? So visual recognition is supported by a processing stream called the ventral visual stream. So as light gets reflected from objects in the world, an image of that object is formed on your retina, and then information is projected from the retina through the optical tract to the back of the brain, where the first visual area, V1, resides. Then information is transmitted to a series of visual areas till it reaches the ventral aspect of the temporal lobe in this region shown here in yellow, which is called VTC, or ventral temporal cortex, and percepts emerge. Importantly, VTC is not a homogeneous piece of cortex, and in fact, it contains specialized regions for recognizing a handful ecologically relevant categories. For example, regions shown here in red on the fusiform gyrus are specialized for processing faces, the region on the occipital temporal sulcus that are specialized for processing limbs. There are other regions in the occipital temporal sulcus that are specialized for processing words, and there are regions in the collateral sulcus that are specialized for processing places. But we're interested to understand how these regions develop during childhood. And one drivers that might affect this development is that there are different varying visual demands for different categories. So while you might see faces and hands or places early on and frequently during infancy, some category of stimuli such as words and reading only become important and frequent during childhood development. And this later development of reading has led scientists to propose different ways in which the brain might develop. So if this is a schematic of some piece of cortex in 5-year-olds, maybe some of it already has domains for faces and words and for faces and limbs, but some of the cortex is not fully developed and shown here by these white voxels. And as you learn how to read, this develops new selectivity, for example, to process words. Another hypothesis has been suggested is this competition hypothesis between word and face representations. So in order to recognize faces or to read words, you have to look at them or foveate on them. So it might be that faces and words are competing on foveal territory in ventral temporal cortex. And this theory suggests that as you become better with reading, your activation for words will increase. And at the same time, this will occupy territory that was previously used to process faces. So in order to test these hypotheses, it's important to collect longitudinal data in children as they learn how to read. So I'm going to describe a study in which we collected longitudinal fMRI data from children. They were initially 5 to 12-year-olds when the study began, and we collected data in them over a course of up to 5 years. So I'm going to show you data from 29 children over 129 different sessions. So in each session, they come and they participate in an fMRI experiment. In the fMRI experiment, they see stimuli from many categories, like the ones that they showed before. And basically what we're interested to see is how selectivity for these categories develop during childhood. Okay, so I'm going to focus on this part of the brain called lateral VTC, which is shown here in green. And the reason I'm going to focus there is this is where the foveal representations reside. And the first thing that we're interested to look at is how does the volume of selective activation, or how many increases as over a function of age. And we're going to do that for each of the 10 categories that the subject sees. So in this scatter plot, each session is a dot, and the sessions are colored by participant. As you can see, consistent with the first hypothesis, we see an increase from age 5 to age 17 in the amount of voxels in the brain that process words. Interestingly, if we look at in the same participants, there's also an increase in the amount of cortical territories that's devoted or selective for processing faces, for example, child faces. But to our surprise, we also found decreases in selectivity. Specifically, we find a significant decrease in the volume of activation that's selective for limbs from age 5 to 17. So we can quantify these developments using a linear mixed model shown here with a red line. And then what we're interested in is looking at the slope of this model, telling us if there's an increase or decrease in selectivity. And we can summarize this for all categories that we showed our subjects. So you can see that, as I showed you before, we find a significant increase in the selectivity to words, but it's constrained to the left hemisphere with no increases in selectivity for numbers. For limbs, we find a decrease in selectivity bilateral, but it doesn't extend to body parts. For faces, we find significant increases for both child and adult faces in both hemispheres. And for the other categories, we don't really find a lot of development, except maybe for a slight development for houses in the left hemisphere. So we see this differential development of category selectivity across lateral VTC. Of course, because we have a longitudinal study, we can see what happens in these regions as they grow. So let's zoom on VTC of one individual child, and this is this child's initial word selective region. And with development, this region is now growing, and this is the end region shown in the black contour. And what is really interesting to see is what happens to the activation in this region that I've colored now in red, which is the emerging word selective region, which was initially not selective to words and later on became selective. So to do so, we look at the amplitude of response for each of the 10 categories. And here I'm showing you this mean response across 5 to 9-year-olds. And you can see that initially this region is not really selective because it shows similar responses for words, numbers, and limbs. So what happens across development? Do responses to words increase, or responses to other stimuli like limbs or numbers decrease? So I'm going to show you the data from teenagers in darker colors. So first we find a significant increase in the response to words, but we don't find significant decreases in the responses to limbs or numbers or to other categories. So here it seems that selectivity is increasing, mostly due to increased responses to words. We can do a similar analysis looking at the region that was initially limb selective. So this is the first session of another child showing the limb selective region, and across development, it actually shrinks. So the end region is now smaller. And again, we can see what happens to these voxels that I'm coloring now in red that were initially selective to limbs and lost their selectivity. So if we look at the mean responses in children, we can see that this region is indeed limb selective in children because it shows the highest responses to limbs. So what happens with development? First of all, we see that there is a significant decrease in the response to limbs as we lose this limb selectivity. And in addition, we see some trends for increases in the responses to words, as well as increases in the responses to faces. So basically we see changes to all of these three categories. So in order to test the predictions of these hypotheses directly, it is interesting to see if we can track the relationship in the changing selectivity to words, faces, and limbs within individual children. So let's consider this emerging word selective regions. So what we're going to measure is a relationship between word selectivity on the y-axis and face and limb selectivity shown on the horizontal axis. And what we're interested to see is the change in selectivity between each individual child's initial to end session. So each child is going to be represented by an arrow. So if we go back to the predictions of the competition hypothesis, this hypothesis predicts a trajectory that looks kind of like this, where initially these voxels have face selectivity or have a positive selectivity for faces, and as word selectivity increases, they lose their face selectivity. So how does the data look like? The data actually does not look like the prediction. While we see, as expected, this increase in responses to words, so all the arrows are pointing upwards, we actually start from a region that has negative selectivity to faces, meaning that it's not face selective to begin with. So to better visualize how things are changing with respect to limb selectivity, I'm just going to take the same data and rotate the plot. So now the limb selectivity is in the front and the face selectivity is on the side. And when we plot the data in this view, you can see immediately that this region has initially limb selectivity, and basically as word selectivity increases, limb selectivity decreases. And we find this effect in 27 out of 29 children in which limb selectivity is recycled into word selectivity. So this is in the emerging word selective region. We can do the same kind of exercise in the face selective regions. And again, we can see that in a similar pattern where limb selectivity in this emerging face selective regions is recycled to face selectivity as children develop. So this is really interesting because it seems that this data suggests that as face and word selectivity develop, the recycled cortical territory that was previously used for visual processing of limbs in younger children. So far I've seen shown you the mean responses across regions, but it's also interesting to consider what happens in development to distributed patterns of activity across ventral temporal cortex. And this is interesting for two reasons. First is because in adults, a lot of research has shown that these patterns carry category information. And in fact, by looking at these patterns of brain activation, you can decode what subject sees. And second of all, you get these distinct patterns also for categories that don't have a category selective regions, for example, for cars. So next we ask, what is the longitudinal development of distributed BTC responses to visual categories? So basically what we're going to do is we're going to measure the distributed responses to each of our stimulus category. So for example, this is a distributed response in lateral VTC to word stimuli. And we can do this exercise now showing the children a different set of word stimuli, and we get a different distributed pattern. And what we're interested in to look is how reproducible these patterns are across different stimuli set for the same category. And you can see, we measure this with a correlation, and you can see the correlation is positive, indicating that these patterns are very similar across different stimuli of a category. So this is true for words, and we can do it for each of our categories. For example, you can see that the patterns of response for two different sets of face stimuli are also positively correlated. In addition, we are also interested in looking how similar are these patterns for different categories. So we're going to measure this correlation, and you can see that this is actually a negative correlation, and which basically indicates that these patterns are actually very dissimilar for different categories. And of course, we're going to do this for all pairwise comparisons between distributed responses to our 10 categories. And this is summarized in what we call the representational similarity matrix. So this is the average representational similarity matrix of right lateral VTCs in 5 to 9-year-olds. And what's immediately obvious is that there is a categorical structure in the brains of 5 to 9-year-olds where patterns are more similar among items of a category and dissimilar for items of other categories. We can also use multidimensional scaling to project this similarity matrix onto axes that explain a lot of variance. And when you look at it this way, you can see that some categories such as faces and bodies are well separated from the rest, but other categories are pretty overlapping. For example, words, number, corridors, and houses are pretty overlapping, so they're not well separated. So to quantify the development of these distributed responses, we're going to use a metric called distinctiveness. And basically, distinctiveness quantify how similar are patterns within a category minus the similarity of patterns between categories. And this basically is a metric that tells us how reproducible patterns are within category and how different they are across categories. So we're going to do this analysis for each RSM in each of the 129 sessions and for each of the categories. So for example, I'm going to show you graphs like this, that the x-axis is age and the y-axis is distinctiveness. And one hypothesis is that across development, these patterns might become more distinct. So we might see an increase in distinctiveness. So if we measure this across our participants, we see that for child faces, we have increases in distinctiveness from age 5 to 17. However, for other categories, such as limbs, we find a different patterns where we actually see that the distinctiveness for limbs actually decreases. And again, we're going to summarize these results with a linear mixed model and measures a slope testing for each of the 10 categories. So for faces, we have an increase in distinctiveness, and for limbs, we have a decrease in distinctiveness. And we also find in the right hemisphere increases in distinctiveness for numbers and houses. And in the left hemisphere, we see increases in distinctiveness for faces, for words, and for houses, but not a significant effect for limbs. So given this heterogeneous development of distributed responses, you might wonder what is the net effect on the representation of children's brain. So we can see this by comparing the RSMs as a representation of categories in 5 to 9-year-olds to that of 13 and 17-year-olds. And you can see that the net effect of this development is increasing of the category representations, which become more dissimilar, more distinct in the teens, and especially the representation of words and numbers. We also repeated this analysis when we restricted it to the selective voxels, and we see a similar pattern. However, when we do this analysis in the non-selective voxels of lateral VTC, we don't see a categorical structure or a development, even though they constitute the majority of voxels of lateral VTC. So given that we found a lot of evidence, both from univariate and multivariate analysis of development of category selectivity, we wondered if there is a behavioral ramification. And specifically, we hypothesized that perhaps as these patterns become more distinct, that would lead to better recognition of the relevant category. So to test this hypothesis, we measured the relationship between reading accuracy as a function of distinctiveness for words. And we find a significant and positive relationship. And this relationship is significant also when we factor out age. Likewise, when we look at the relationship between face recognition ability and the distinctiveness of the pattern across VTC for faces, we also find a significant and positive correlation, which is also significant after factoring age. So this suggests to us that as these patterns become more distinct, there's better readout of category information and which support better recognition of stimuli from these categories. So to conclude, our data shows the first evidence of recycling of category selectivity in ventral temporal cortex during childhood development. And specifically, we find that limb selectivity is recycled to word and face selectivity. And together with this recycling, we also see increases in the distinctiveness for distributed responses to words and faces, but decreases in the distinctiveness of distributed responses to limbs. And these developments are important because they're linked with improved perception of faces and word. Now, Finally, you might wonder why there might be this recycling in the first place. So we hypothesize that this recycling during childhood might reflect an adjustment to the visual demand and frequency of visual stimuli that are important for children. So for example, babies look more at hands and faces, but toddlers look more at babies look more at faces and hands, but toddlers look more at hands and faces when they learn to manipulate objects. And we hypothesize that later in childhood, other visual stimuli and tasks such as words and reading might become more frequent and important, which might lead to recycling of cortical representation for the more salient stimuli. And of course, this hypothesis needs to be tested in future research in which we could actually measure the visual diet of participants together with their brain development. So thank you for your attention.
Thanks so much, Kalanit. Great talk and time for questions. I was wondering if it's feasible or planned to do these types of analyses in situations where kids might have reading impairments or visual impairments, and to see if some of these brain regions have sort of delays in developing or things like that.
I think this would be really interesting because one thing that you could look, for example, in people that have reading disabilities is how do these, if they develop less fast or at a different rate, or maybe the patterns of words and faces aren't as distinct. I think it's also really interesting to look at other kind of disabilities. So one thing that might be interesting is actually deaf people, because deaf people use sign language. So the hands have to be probably visually very important for deciphering language. And it's actually a different language than written language. So that might be a population to examine also, because again, they have different visual demands compared to typically hearing people, basically.
Got it. Okay, Professor B.
Thank you, very nice talk. Yeah, I think your response answered one of my questions, is why you think the limb selectivity is recruited. Like I can imagine why face, because these voxels may be good at processing foveal information, like high resolution. So images, that's good for written words. But why do you think the limb, it's just because these voxels are not used as much, or they have something to do with the functionality of these voxels to begin with?
So first, I think these are all in territories that has foveal vision. Both the words, the bodies, and the limbs and the faces are all foveal territory. So I do think it's something about the foveal territory being recycled. And when we measure the foveal bias, we actually see that it develops in these face and word selective region across childhood. It is actually enhanced. I also think that the way we look at the world and that's what I call it, the visual diet, is changing across childhood development. So if you think like a 1-year-old or pre-verbal child, they use a lot of gestures to communicate, right? You teach the baby to look, hey baby, and you point, and the gestures are really big and prominent. So there's a lot of effort. You know, I remember my kids learning how to ride their bike. They look at their legs when they're looking at the bike, because they're not really sure that their legs move without them looking at them. So there's a lot of visual guided actions that requires you to learn to use your limbs. But I think as we become more sophisticated, some of this communicative information and also our motor skills don't really require us all this visual attention on our limbs, but instead other stimuli become really important. I spend hours a day like looking at words. I mean, this is really what I do. And I think that's what I really think it's about what we're using our vision for.
Right. So can I follow up on that? So that's my wild alternative thinking. What these voxels are, what we use our visions for is because words are used for language. So one property is a visual characteristic. The other is they are used for communicating with our language cortex. Do you think it's possible that actually the gesture is also communicative? So maybe these voxels are better at communicating with these frontal, temporal, parietal language areas. So if you look at connectivity pattern, so that's the final question is the lateralization. Maybe I missed it. So the word is more on the left, then the faces become the right. So is that transition related? Thank you.
Is that what related?
That lateralization of the two related.
So first of all, a couple of things. So the lateralization is really big, and we see it consistently across studies, that the words really develop in the left hemisphere, and the faces develop bilaterally, but more so in the right hemisphere. So faces are less lateralized. We have a paper that we just put on Bar Archive. I think my student put it actually yesterday, where we actually look at the white matter connections between ventral temporal cortex and the rest of the brain. And it's kind of interesting because these connections, when we look at them, they're really scaffolded by the cytoarchitectonic partition and not by category selectivity. But we see some changes in the patterns of connectivity, but that seems to be much more specific to the words. So some people think that the language pathway is very hardwired, but it doesn't seem that it's so different. So there's a lot of similarities in the connections of faces and words to the front of the brain. And it's actually the posterior versus anterior face and word areas that have this different pattern of connectivity. So yeah, that's my answer about connectivity.
Okay, Gary, you had a question. Do you want to ask about dyslexia?
Yeah, I just wondered, I mean, you showed that face recognition performance correlated with distinctiveness of representation. I wondered if you had any dyslexic kids and whether their distinctiveness was especially low compared to good readers. I mean,
So,
maybe you mentioned that, I didn't see.
Yeah, so we only measure typically developing children, and we actually don't recruit people that have any sort of reading disabilities. That's kind of like an exclusion criteria for our studies there. But I think it's a really good question. That's an obvious thing to ask from our results. So other labs at Stanford, such as Jason Needman's lab, are looking at dyslexic kids and also at interventions. They have some of the stimuli that we use in our localizer, they're using a subset of that, and they would be able to answer your question with the data that they have been collecting.
Okay, sorry, my question was obvious.
No, it's not obvious at all. It's a good question. Sorry, I didn't mean to be obvious. It's a good question that seems like a next step that we should do, but in my lab, we're not recruiting.
You're looking at developing kids. How do you know they're dyslexic before they learn to read?
Well, for 5-year-olds, we don't know. And we've had subsequently 1 or 2 kids that were identified as dyslexic and they were excluded from the study, basically.
Oh.
We don't have enough to compare, basically.
Yeah.
Yeah.
Ask a question. Hi, we're using to your talking about Univ, so it's my student's computer. So I'm standing here from Big University. I'm interested in autism, and there, I guess the hypothesis you started with was particularly appealing because a lot of these children with autism, they're very good at reading words and numbers, but not good at faces. Can you discuss or speculate whether you think they have a problem with transition, or somehow do the word, or for in these cases, maybe do the words compete with the faces, or they just don't do the limb to face transition?
Again, I haven't measured autistic children, so I can only speculate here. There's research on autism. Some of it is controversial, but there's some old paper from 2004 from Dalton who suggested that children with autism might not look the same way at faces as typical participants. And specifically, usually when you look at faces, you look at the center of the face, which is kind of in between like your eyes, because that's basically putting the receptive fields on where the features are for face recognition. And there's some research that suggests that the autistic people would look more at other features like the mouse. So they're not looking in the same way. So I will speculate and suggest that if their looking pattern is consistently different, that would affect their face representation and would affect this recycling hypothesis. Of course, I'd be very happy if somebody would actually measure it and either say if I'm right or wrong. Either way, it would be interesting for me to know.
Oh, thank you very much.
Any other questions? Okay, if not, thanks so much, Kalanit, for a really engaging, exciting talk, and thanks, Andre, for an amazing talk as well. Good to see everyone, and have a happy Chinese New Year, and we'll be back February 7th and 8th for more talks. Thanks, everyone.
Thanks,
Thank
Andre.
you.
Great. Thanks, everyone. Happy Chinese New Year. See you in 2 weeks.